Okay, today we're going to walk through Google Genomics Codelab written in the R programming language. You can find this Codelab and much more in the Google Genomics organization on GitHub. Let's go into the Codelab repository. And let's clone this repository so that we can work through it ourselves. So I'm going to copy the clone URL, and today I'm going to be using RStudio, which is a really fantastic integrated development environment for R, and it integrates nicely with GitHub. So in RStudio, we're going to go to File, New Project, Version Control, Git, and paste in our repository URL, and hit the Create Project button. And this is going to bring this down to our local machine. But for now, let's go back to GitHub to look at the content here. So what we're interested in is the Codelab in the R directory. You'll notice that there are some prerequisites, so you're going to want to walk through these first to get your R environment set up to run this Codelab, but we're not going to go through them right here. The Codelab we're going to run today is a data analysis using Google Genomics. We're going to use a couple different modalities to work with the data: cluster computing using Apache Spark, large-scale analytics with Google BigQuery, and using the Google Genomics API directly to bring data into R and work with BioConductor. So, as I mentioned, you want to do the prerequisites first, but now let's go ahead and look at the rendered version of the final analysis. This Codelab uses reads and the phase 1 variants from the 1000 Genomes project. The VCFs comprising this dataset are over a terabyte when uncompressed and provide information about 39 million variants for 1,092 individuals. For more detail about how this data was loaded into Google Genomics, please see Google Genomics public data. All the details are there. So as I said, what you're looking at here is a rendered version of our analysis. We can read it from start to finish here on GitHub, but since it was generated from code, we can also run it chunk by chunk or line by line in R. First, we'll work at scale and analyze the full dataset. Then we'll drill down to a specific region of the genome. So suppose we have a new dataset. One of the first things we might do is a visualization on the structure in our dataset. Let's start by projecting the relevant data into two-dimensional space by performing a principal coordinate analysis. This is an n-squared analysis where we count the number of variants shared by each pair of individuals, create a similarity matrix from the data, center it, scale it, and compute the first two principal components for each individual. With just a few clicks, it's easy to spin up an Apache Spark cluster on Google Compute Engine and run this analysis. But we're not going to do that today. See the Spark Codelab video for more details as to how to run this on many machines in parallel to get the analysis done quickly. In this example, we're just going to read in previously computed results. So using ggplot2 to visualize the results, we see quite distinct clusters. Now let's pull in the supplementary information we do have on these samples from Google Cloud Storage. For the 1000 Genomes dataset, we have gender and ethnicity. So adding ethnicity to the plot, we can see that it appears to strongly correlate with the clusters. So what does this look like when we run it from R? So I'm walking down that same tree again. I'm going to the R Markdown file that was used to generate the Markdown we were looking at GitHub. Opening that up. And you can see that we've written this in a literate programming style, interleaving narrative text with code. So let's go ahead and start running this. The first thing we want to do is ensure that our working directory is the same one where the Markdown file resides, the R Markdown file. And in RStudio, you can just click on that, which is pretty handy. But we have the command right here if you're using R, not RStudio. And now we're going to read in the results of that Spark job and pull in ggplot2 and kick off our plot. Oops. And we're going to read in those results. and kick off our plot. And this just looks like the one we were just viewing. We're going to go ahead and now read in that file from Google Cloud Storage. And we're going to use the dplyr package to join it with the results from the Spark job. And again, replotting those results. And now we can see that the clusters are pretty clear. So let's go back to looking at this on GitHub. Next, let's analyze a different aspect of this data by examining the variant counts for each individual where one of the alleles differs from the reference and where both alleles differ from the reference. This is an order n analysis. We're going to use Google BigQuery to do this. First, we'll use the Google BigQuery web UI to run this query. So I'm going to copy the query and paste it into the BigQuery web UI. Pull it down so we can see it. And click on the Run Query button. Since BigQuery uses a columnar data store, it scans only the columns referenced by the query. In this case, it's going to be about 1 terabyte of data scanned, and this query runs typically within 15 to 20 seconds. And here are the results. We have the counts for each individual in the dataset. Now we can also run this exact same query from R using the BigQuery package. So let's go over here and take a look. So first off, we want to load the BigQuery package. And we want to set which project, which Google Cloud Platform project we're running this as. In my case, it's Genomics Public Data, but you'll want to put your own project ID here. And then I have just a little helper function that makes it easier to run these queries. So here's the first one that I'm going to run, which is the one we just saw in the web UI. So my little helper function just catted it out to the R prompt so we could see it. And now we have our results back. And results, they're just a data frame in R, which is pretty great. All right. All right. And let's again join these results with the sample information that we have and redo our plot. By the way, I should say in RStudio, you can just highlight commands and use command return on a Mac to send those commands down to the console. Okay, now that we've run some analyses across the entire genome, let's shift gears and zoom in. Suppose we're interested in examining variants within the BRCA1 gene. We might run our principal coordinate analysis a second time, zooming in specifically to this region within the genome. Again, in this example, we're going to read in previously computed results, but since the amount of data over which we are computing is much less, it's feasible to run this Spark job on a local machine in just a few minutes. So let's switch back to R. Okay, we're loading in the results from that smaller job and plotting them. Okay, and we see that the clusters are quite different in structure this time. And again, we're going to join with our sample information using dplyr and then replot. Now this time I put gender on there, and we can see that it has no bearing on this clustering. So let's instead apply ethnicity. And we can see that ethnicity does appear to explain some of the difference in clusters between the top and the bottom of this plot, but not the difference between the clusters on the left and the right. So what could that be due to? It would be interesting to find out. So let's split these individuals into two groups based on their position relative to the origin of the first principal component. So first off, I'm going to mutate the data frame to add a new column as to whether the sample is case or control based on its position relative to the origin. And I'm going to plot that again, showing case or control with shape, just so you can see how the data was split. So you can see case and control, opposite sides of the plot. Okay. The next thing we want to do, let's get back to where we were, is perform a simplistic GWAS query on the BRCA1 variants to retrieve a ranked list of the variants that appear to differentiate these groups. It's doing a simple chi-square test on the variant count differences between case and control groups, and it's a pretty long query. So let's go back to R and run this. And here you can see that we're getting the case sample IDs, and we're dynamically adding them into our query at runtime. And here's that query printed out, and our results are back now. And there they are. Okay, so these variants at these positions are the ones that most differentiate the two groups we see in our plot. Now, why is that? What's interesting about these variants? So let's keep going. Oh, and I want to add that even though this query ran over a small region of the genome, with a minor change to the SQL, we could have run this same GWAS query over all the variants within a much larger region, over an entire chromosome, or even the full dataset, returning the ranked list of variants that differ between the two groups. Yes, that's right. We can use BigQuery to run 39 million chi-square tests and return the top ranked variants. This is a pretty big hammer. You might do this to get the top variants and then re-examine those identified variants with a more sophisticated and nuanced statistical analysis. So now let's use the Google Genomics R client to retrieve the full records for the top 20 variants returned by the query. So we're going to load it in. And then we want to authenticate. So you want to edit that to the path to your own client secrets file. We're going to grab the top 20 of those results that we got back from BigQuery, and we're going to get them one by one. Okay, so we got 20 back, and you can see these are the field names, top-level field names for the objects we got back. And it's a nested list. So the variants at the top are at the top level, but the calls are nested underneath them. So for our first variant, we have calls on 1,092 individuals, which is common across the 1,000 genomes data set. Every variant says something about all the individuals in the data set. And what fields do we have for the calls? You can see we have call set ID, call set name, genotype, phase set, genotype likelihood, and so forth. And let's go ahead and just look at one of those calls. So we have the name of the individual, the genotype 00, phase set, and the likelihood. By default, these are returned as an R-list object, but we can also convert this data to BioConductor data types, such as the G-ranges type. So let's do that now. All right. This allows us to utilize the various BioConductor variant annotation packages, which is wonderful. So let's go ahead and load those in. We're going to load in the variant annotation package, HG19, and the mapping of genes to the HG19 reference. So let's locate the coding variants let's locate any coding variants that were in that set that was returned by BigQuery. Okay. And we can see that we've got lots of great identifiers that we can look up with additional annotation databases from BioConductor. But let's move on to predicting the impact of these variants upon the coding. All right, so you can see that we've located the coding variants and predicted their impact upon the coding. There's a ton more that we can do with BioConductor, but we haven't actually yet answered our original question about the clusters. So a question for our users who have much more experience in this domain: what should we examine next to determine the potential explanations for the clustering we saw over here for BRCA1? Send pull requests or GitHub issues. All ideas are welcome. So lastly, let's dive in even a little further. What if we, for example, want to see the data used for a particular variant call? We can also retrieve the reads from the reads API for a given sample. And here, you can see we passed in a converter to just automatically convert them to a BioConductor G-alignments object right away. So here are the reads as G-alignments. And we can load the ggbio package. And plot our pileup and our coverage. In summary, this Codelab ran all from the R prompt, and we were able to exercise both large-scale and small-scale data analysis using cloud-based infrastructure. Thanks.